Hi, I'm Kevin Biglin. I'm an associate professor at the University of Rochester, and I'm going to be speaking about experimental therapies in Huntington's disease. My disclosures are, just to give you an outline of what I'm going to be talking about today. First, I'm going to be discussing why we do clinical trials, what's the importance of clinical trials, and what are the mechanisms that we use to show that therapies are beneficial. Update of some recent trials in Huntington's disease, a brief discussion on future directions, and then there'll be a slide for resources to learn more information about Huntington's disease research. So why do we do clinical trials? We do clinical trials because it's the accepted method of determining unequivocally whether a treatment is effective. What is a clinical trial? It's a prospective study comparing the effectiveness and value of an intervention against a control in humans. So what does that really mean, actually? Well, it's prospective, so we're collecting the information as it occurs. We're not looking back at time and see how, if somebody was on a particular treatment, how did they do on that treatment in the past. It's really collecting the data as it occurs going forward. And interventions can be a variety of interventions. Experimental treatments such as medicines most commonly we think about, but certainly surgery, stem cells, even exercise could be a form of intervention that we'd be interested in looking at in Huntington's disease. Against a control group, so a group that's not receiving the experimental treatment. Traditionally, we think about a placebo, a sugar pill. Sometimes in surgical interventions, we'll think about sham surgery, so individuals will actually not receive the actual surgery, but close enough, so to speak. Or it could actually be an existing or already proven treatment. So if we were to have treatments that were effective for Huntington's disease, we wouldn't necessarily want to keep those away from individuals with the disease, but if we're interested in looking at it, whether a treatment is more effective or as effective as the existing treatment, the control group could actually be that already proven treatment. And finally, we're talking about humans. We're not talking about worms, flies, mice, rats, or monkeys. We're really interested in whether this treatment has a meaningful benefit in individuals with the disease. In terms of the drug development process, particularly the drug development process, we often think of 3 phases of drug development, and I'll be speaking a little bit about trials that have been in all 3 phases, so I think it's worth understanding the terminology. Phase 1 is what we think of as a safety and tolerability study, generally in healthy individuals, though occasionally we will look at phase 1 studies when we're looking at safety, toxicity, pharmacokinetics in individuals with the disease in question. These are relatively short duration studies, small numbers of patients, sometimes lasting hours, sometimes just lasting days. A phase 2 study really is the next step. Once we have a better understanding of the potential therapy in terms of its pharmacokinetics, its potential toxicity, its potential safety concerns, we're looking at broader safety and tolerability issues. What are maximally tolerated doses? These are generally done in individuals with the disease in question. We may be interested in identifying the dose that we want to study further in definitive efficacy studies. These tend to be longer duration studies lasting weeks to months with larger numbers of patients. And then finally, the phase 3 study, which is really the definitive efficacy study. These are long duration studies involving large numbers of patients, sometimes thousands of patients, and require a lot of time commitment and financial commitment to show that a treatment is effective. When we think about experimental treatments, I like to think about 4 categories of potential therapies in Huntington's disease: the symptomatic treatments, neuroprotective treatments, restorative therapies, and preventive therapies, which are really maybe a subset of the neuroprotective type of neuroprotective treatments. So a symptomatic treatment is a treatment that improves the symptoms of the illness but does not affect the underlying disease itself. So symptoms may be improved, but the disease continues to progress, underlying disease continues to progress, and the benefits of the treatment are only temporary in the setting of ongoing neurodegeneration and progression of illness. In Huntington's disease, this has been a very hot area of research. Recently, tetrabenazine was approved in the United States for the treatment of chorea in Huntington's disease. This slide shows individuals randomized to tetrabenazine, escalating doses as needed, versus a placebo sugar pill group. And you can see that the individuals on in the dashed line who received placebo really had no benefit in terms of chorea. Here, a negative change is an improvement in chorea or involuntary movements, whereas the solid line is individuals treated with tetrabenazine. You can see a marked improvement in chorea associated with this treatment. However, at 12 weeks, when everyone came off the drug, you see that the patients who were treated with tetrabenazine really go back to where they were before. There's no sustained benefit to the treatment. Treatment's purely symptomatic, purely results in a reduction in the involuntary movements in Huntington's disease, and is classically a symptomatic treatment. And is the first treatment approved in the United States for the treatment of Huntington's disease. There are a number of potential symptomatic therapies currently being looked at for the treatment of a variety of symptoms in Huntington's disease. Here we're looking at a drug called prodopidine, or ACR16, which I find always easier to pronounce. This is classically thought of as a dopamine stabilizer medication, what does that mean? I'm not sure the manufacturers even know what that means. However, the idea is that in the presence of a hypodopaminergic state, you're enhancing dopaminergic transmission, so we think potentially that some of the fine motor control or motor control in Huntington's disease may be impacted by a relatively low dopaminergic state. In that state, we can improve motor control, voluntary movements. In a hyperdopaminergic state, which we think is associated with the involuntary movements of Huntington's disease, you can dampen that response. And so the idea here is that you might get an improvement in voluntary motor function while simultaneously seeing a reduction in the involuntary movements. So unlike treatment like tetrabenazine, where you have a dramatic impact in the involuntary movements, there's some suggestion that that might be a cost of voluntary motor function. So on the left slide, this was the original pilot phase 2 study that was conducted in Scandinavia, a small number of patients, 58 patients, who were randomized to receive 50 milligrams of ACR16 for about 4 weeks. And you see that even after 2 weeks, there's a marked improvement in what they call the modified motor score, which is a subset of the Unified Huntington's Disease Rating Scale motor section that looks primarily at voluntary motor function, and that persisted at 4 weeks. And in this study, actually, they did see an improvement in chorea as well, but not an improvement in dystonia. There were 2 larger phase 2 studies, phase 2/3, safety tolerability early efficacy studies. One conducted in Europe, this is the MERMAID study, and one in North America, this is the HEART study. In the MERMAID study, individuals were randomized to receive either 45 milligrams per day or 90 milligrams per day of ACR16. In the HEART study, they looked at a wider range of doses, 20 milligrams, 45 milligrams, and 90 milligrams. And individuals treated with ACR16 seem to have a trend towards improvement in the modified motor scale, again, this subset of the Unified Huntington's Disease Rating Scale that primarily measures voluntary motor function. However, these improvements did not reach their pre-specified threshold for statistical significance. However, when they looked at the total motor score, so looking at all components of motor function in Huntington's disease, there was a statistically significant improvement in both these studies. So based on this information, the FDA did not feel that the benefit of the drug warrant approval at this time and are requiring additional study of the medication. However, it looks like a promising compound to improve motor function in Huntington's disease and certainly warrants further study. Moving on to cognitive function in Huntington's disease, certainly treatments for cognitive dysfunction in Huntington's disease are a major unmet therapeutic need. While there have been some studies, small studies, looking at acetylcholinesterase inhibitors to see if they improve cognitive function, they haven't been overwhelming at this point. Recently, a compound dimebon was evaluated in a phase 2 study. This is a novel antihistamine that's been explored in Alzheimer's disease, unfortunately without benefit, more recently in Huntington's disease. And the initial phase 2 study suggested that there might be some benefit in terms of cognitive function, here measured by the mini-mental state examination. And again, individuals in the dashed line here are treated with the experimental treatment dimebon compared to the solid line individuals on placebo. And you see that there's some suggestion that there might be around a 1-point improvement in the Mini-Mental State Examination score with Dimebon. Again, this was encouraging, and people were interested in looking this further. So a phase 3 study was completed, both in Europe and North America, by the Huntington's Study Group and the European HD Network. 403 individuals enrolled, randomized to 20 milligrams of Dimebon 3 times daily versus placebo for 6 months. And unfortunately, there was no improvement with this treatment, either on Mini-Mental State Examination or the Clinical Global Impression by a blinded rater. This is obviously unfortunate. This is, as I mentioned earlier, a major unmet therapeutic need in Huntington's disease. Treatments that can improve cognitive function are sorely needed. Next, I'm going to talk about neuroprotective therapies. To use the cliche, this is the holy grail of therapeutics in Huntington's disease, as well as other neurodegenerative disorders. Just a brief definition, neuroprotective therapy is a treatment that favorably modifies the underlying disease process, ultimately resulting in a slowing of neuronal cell loss. Clinically, individuals' progression would slow. If the treatment was very effective, it could ultimately be halted, resulting ultimately in a longer duration of functional independence for individuals with Huntington's disease. This slide that is widely circulated is just an example of the natural history of neurodegenerative disorder, and I think it's useful to think about this issue. So the yellow line here is basically some measure of neuronal function or neuronal cell density. The red dashed line here is a measure of clinical symptoms of the disease. At some point in the disease starts, probably in Huntington's disease very early on, potentially even in utero. At that point, you start to develop neuronal dysfunction, neuronal cell loss. At a certain point, that neuronal dysfunction or loss of cells is significant enough that people begin to develop symptoms. These symptoms may be very subtle initially, and this process may occur over many decades until they meet a threshold of clinical symptoms that a neurologist or somebody else says, you know what, I think there's something wrong here. I think you may have a disease, in this case, Huntington's disease. The goal with a neuroprotective therapy currently is we've been looking at neuroprotective therapies primarily in individuals after they've been diagnosed. And you can imagine that if you can slow or reduce the severity of that slope of the yellow line, that you might have some impact on clinical symptoms and clinical function. When we talk about preventive therapy later, the idea is to intervene in a similar way prior to the diagnosis of disease. As you all know from medical school, this is Coenzyme Q10. There will be no test on this later. So Coenzyme Q10 really generated interest based on a study, the CARE-HD study. And this was a study that looked at both Coenzyme Q10 and racemide, which is a glutamate antagonist treatment, to see if either would have an effect on disease progression in Huntington's disease. The primary outcome of this study was the total functional capacity, which is a measure of function in Huntington's disease on a 0 to 13-point scale, with 13 being normal function and 0 being completely disabled. And you can see on the left slide, individuals treated with Coenzyme Q10, that they're in the dashed line again here, compared to the solid line where individuals not treated with Coenzyme Q10. There is some suggestion around 12 months that these two lines seem to diverge, that individuals treated with CoQ, the slope of the decline seems to be less, and they seem to be progressing at a slower rate. When you look at the actual numbers, CoQ resulted in about a 2.4 change over 30 months compared to 2.74 in individuals not treated with CoQ. This was not statistically significant, though the p-value was 0.15. On the right slide, on the right graph, you see there's really no difference with racemide treatment at all. However, when we looked at kind of corroborative measures, the functional assessment checklist, which measures your ability to do a variety of tasks, ability to work, ability to drive, ability to take public transportation, etc., there was a statistically significant slowing on worsening on the functional checklist with CoQ, about 22% compared to individuals with not treated with CoQ. Again, a corroborative scale called the independence scale, a 0 to 100-point scale with 100 being normal function and 0 being completely disabled, showed a suggestion of some benefit there as well, 18% slowing on the independence scale. This approached significance with a p-value of 0.06. So based on this information, there was a lot of excitement that there is some suggestion that CoQ may have an impact on disease progression in Huntington's disease, and currently there is a large ongoing phase 3 study of CoQ in Huntington's disease, largely based on the results of the CARE-HD study, but using a much higher dose of CoQ. So in the CARE study, they looked at 600 milligrams of CoQ. In this study, they're looking at 2,400 milligrams of CoQ, largely based on a small study that suggested that at around 2,400 milligrams beyond 2,400 milligrams of CoQ, you did not see much increase in plasma levels beyond that. And so that was the maximum dose they looked at here. They've recently completed enrollment, 608 individuals, and we'll be following those individuals for an additional 5 years to see if there's any benefit, again, on the total functional capacity. There's another study of CoQ that we'll talk about in a minute called the prequel study that is really a phase 2 study looking at safety and tolerability of CoQ, as well as the effect of CoQ on biomarkers of oxidative injury in a population of individuals who carry the mutation for Huntington's disease but are not yet diagnosed with the disease, so pre-manifest population. This is my segue into biomarkers in Huntington's disease and how can we utilize biomarkers to improve our identification of therapies for Huntington's disease. So 8-hydroxy-2-deoxyguanosine, or 8-OHDG, for those who are easily tongue-tied, is a measure of oxidative damage to DNA. It's stable in serum, believed to be a useful biomarker of oxidative injury in Huntington's disease. There's some suggestion that most of the 8-OHDG that we collect in the plasma, in the blood, is generated in the CNS, in the central nervous system. In postmortem studies of Huntington's disease individuals, there are elevated levels of 8-OHDG in the brain, in the striatum, where we know Huntington's disease is particularly violent, as well as cortex. In animal models, the R6-2 model also shows an increase in 8-OHDG levels. In addition, other neurodegenerative diseases where we think oxidative injury is an important factor show elevated levels of 8-OHDG, such as ALS and Friedreich's ataxia. So why is 8-OHDG important? If we're interested in looking at therapies that may have an impact on oxidative injury and therefore have an impact on neuronal cell loss and disease progression, it'd be useful to be able to identify those treatments early. So Steve Hirsch and colleagues were interested in exploring creatine as a potential therapy for Huntington's disease. And in a phase 2 study, they showed not only was it safe and tolerable, but they showed it had an impact on 8-OHDG levels. So 8-OHDG levels were actually reduced, suggesting that creatine may reduce 8-OHDG and therefore be an antioxidant that has a meaningful impact in Huntington's disease. Similarly, we've shown that this is true for CoQ as well. And so individuals treated with Coenzyme Q10 also showed a reduction in 8-OHDG levels. This allows us to screen potential antioxidant treatments to see if there is an impact on 8-OHDG and therefore have a potential impact on the disease. Ultimately, our ability to show an impact on 8-OHDG is going to need to be proven to have an impact on meaningful clinical outcomes. So creatine, as I alluded to earlier, was shown to be safe and tolerable in Huntington's disease, was shown to have an impact on 8-OHDG levels, and largely based on that information was progressed to a phase 3 study looking at the efficacy. This is a large study, again, an international study, which I think is ultimately important for the identification of treatments in Huntington's disease. 650 individuals treated with high doses of creatine, up to 40 grams per day, and followed for a period of 36 months. Similar to CARE, the total functional capacity is the primary outcome here, though they're also looking at imaging measures as well to see if there's an impact on imaging outcomes. So the CREST-E study, the study of creatine in Huntington's disease, is ongoing. Now I wanted to switch gears a little bit and talk about, as opposed to antioxidant therapies that may have an impact on disease progression in Huntington's disease, looking at treatment that may actually directly impact the primary mechanism involved in the pathogenesis of Huntington's disease, which is the mutant Huntington protein. I think we believe at this point that the instigating event in Huntington's disease is the translation of this abnormal protein from a expanded trinucleotide repeat in the mutant Huntington gene, that the deposition of this mutant Huntington is somehow a key piece in the development of Huntington's disease. And so a company, Sienna Biotech, is now looking at a compound called solicitat. It's very difficult to pronounce. It's a SIRT1 inhibitor. It has a funny, long numerical name, as many of these drugs do as well. But it's a novel mechanism, and in animal models, it has been shown to selectively reduce the abnormal Huntington protein in the brain. And one can envision that if you could reduce the abnormal Huntington in the brain, that maybe you could impede the progress of disease. In mouse models, on the graph on the right is a Kaplan-Meier survival curve in individuals treated with this SIRT1 inhibitor, and you can see a prolongation of life of about 100 days in mouse years. There's a significant effect on lifespan in these models, motor outcomes, as well as histological outcomes with reduction in Huntington deposition in the brain. So a phase 2 study, a safety and tolerability study, is currently underway in Europe, and there is a first phase 1b study in patients in North America, looking at whether or not there's an impact of fasting versus fed state on the pharmacokinetics of this compound. If you recall, we talked about phase 1 studies being really about early toxicity and pharmacokinetic information. Just a little bit more information about the ongoing studies in North America and Europe. The phase 1b study in the United States is primarily looking at whether there's an impact on the pharmacokinetics of this drug when taken in the fed or the fasted state. This is a requirement of the FDA prior to moving this drug forward into phase 2 and phase 3 studies. And I think given the mechanism of the drug, people are very interested ultimately in the outcome of this compound. In Europe, they've already moved to phase 2 studies, and they're doing a larger study at 18 sites in Europe as a component of the European HD network. 3-month exposure to 2 different doses of this SIRT1 inhibitor. Next, I'm going to talk about restorative therapies. The idea here is that a treatment that either promotes or facilitates the regrowth of new neurons in damaged regions of the brain affected by Huntington's disease. In reality, this would potentially be a cure as disease severity is lessened and people's function improves. The primary approaches that have been taken to this point in terms of restorative therapy have largely been in terms of cell transplant therapies. And Hauser et al. Published this in 2002, where they looked at 7 individuals who received bilateral fetal striatal transplantation in Huntington's disease. 7 individuals, open label, no real control group here, so I think these are important caveats of this study. One of the biggest issues was that there was a high likelihood of adverse events, particularly subdural hemorrhage. 3 individuals developed subdural hemorrhage in this study. 2 of them required surgery. 1 individual actually ultimately died in this study, unrelated to the study. The individual had a heart attack and died. It was an opportunity to do an autopsy and see that the actual tissue transplantation did survive. When you looked at clinical measures, particularly the Unified Huntington's Disease Rating Scale, there was no improvement when you looked at it overall. They did do a post hoc analysis looking, removing 1 individual who did particularly badly in the study, and when they removed that 1 individual who did poorly, they did see that there was a benefit on the Unified Huntington's Disease Rating Scale, even though they were looking at a small number of individuals. They also showed that there seemed to be integration of these transplants in the brain using PET scanning, and this is an example of what that might look like. These are 5 patients, actually in a different study. But if you look in the striatum, you can see the red areas are areas of high activity, And you can see from the before transplantation to the after transplantation, particularly in patient 1, where the arrow is, that there is this increased uptake in the striatum, suggesting that there is some functional benefit to the transplantation. Though ultimately the issues surrounding the ethics of fetal transplantation, the issues surrounding the safety of fetal transplantation in this population really have put a damper on this treatment. But you could imagine that future therapies looking at stem cells or other potential cell transplantation techniques might be of value in Huntington's disease. Finally, I want to talk about preventative therapies in Huntington's disease, and I think as a community we see this really as the way things are going ultimately. We are fortunate in one respect that we have a disease we can identify with 100% certainty prior to the onset of symptoms. So if we could intervene in individuals who are at 100% risk for developing disease prior to the onset of symptoms or the onset of illness, presumably we can delay the onset of symptoms, thereby delaying diagnosis and ultimately delaying functional decline in these individuals. So a preventive therapy is analogous to a neuroprotective therapy. However, we're looking at it prior to the onset of symptoms. We are favorably modifying the underlying disease process. Neuronal cell loss, again, slows, but when initiated prior to the onset of symptoms, we delay the onset or prevent the onset of illness. This figure is taken from Jane Paulson's article on the PREDICT study, which is an observational study looking at individuals prior to the onset of illness. And here we have individuals who are mutation positive. They carry the CAG expansion for Huntington's disease. And the downward facing arrow is a point prior to individuals reaching this diagnostic threshold, whatever it might be. It's traditionally been motor, but it could change in the future, where we intervene with some treatment that has an impact on the underlying disease process. Individuals in a randomized trial, individuals will be treated with a treatment and placebo. And so individuals with the square here are untreated individuals compared to the triangle. These are treated individuals. The rate of functional decline slows or symptom development slows, and people are are diagnosed at a much later time period. And this is kind of the model that we're thinking about when we talk about interventional therapies to delay or prevent the onset of illness. Currently, there has not been any studies that have shown this. That may be obvious. We have just completed a study of coenzyme Q10 in this population. This is the prequel study. This is a phase 2 study looking primarily at the safety and tolerability of 3 dosages of coenzyme Q10. The rationale here was that individuals in an earlier study who were otherwise healthy seemed to be more poorly tolerant of coenzyme Q10 than individuals who were ill or already had manifest Huntington's disease. This suggested that in otherwise healthy individuals, there may be a differential tolerability of treatments in the well individuals compared to the diseased individuals. This study was a small study, 90 individuals, followed for about 20 weeks, randomized again to 1,200, 2,400, and 3,600 milligrams of CoQ, looking at safety and tolerability, and as alluded to earlier, looking at the impact of coenzyme Q10 on 8-OHDG levels. These results will be available soon. There's a lot of excitement in Huntington's disease research about means for preventing the translation of the abnormal Huntington protein. The most noteworthy approach to this is using RNA interference techniques, and this has shown a lot of promise in a variety of animal models. So all different animal models using different techniques to silence or reduce the translation of the mutant Huntington. They've shown benefits, histological benefits in these animal models, as well as clinical benefits in terms of motor function primarily. This slide shows a simplified mechanism of how one approach to RNA interference may work. We take small non-coding RNA sequences which are targeted towards the Huntington mRNA. In theory, you could potentially use single nucleotide polymorphisms to target specifically the mutant Huntington protein. These small non-coding RNA sequences will bind to the Huntington RNA, leading to its cleavage, degradation, and a reduction in the translation of the abnormal protein. Presumably, a reduction in translation of abnormal protein would result in a reduction in neuronal dysfunction, reduction in neuronal cell loss, if done early enough, potentially prevention in the onset of symptoms and diagnosis of Huntington's disease. In the section A here in the middle row, we're looking at a reduction in Huntingtin protein in red in those individuals treated with this RNA interference. On the right side, we're looking at functional improvements in individuals treated with these RNA interference techniques. Unfortunately, there's still a lot of unanswered issues related to RNA interference. There are two major approaches to them. One is called a short hairpinned RNA interference, and one is a small interfering RNA. They both have different benefits and potential problems. The what they call shRNA has the potential to be incorporated into regions of the DNA outside of the Huntingtin protein, thereby disrupting other genes that are not intended to do so. Having said that, it tends to be the approach that results in the most long-term benefits in silencing mutant Huntington's disease. So there's a trade-off there. The siRNA, or the small interfering RNA, while more short-acting, are more likely to be degraded, are less likely to cause distant effects, but might require repeat infusions of the RNA compounds to maintain the benefit over time. And there really is a lot of unknown about how often you would have to do this, how much do you have to reduce the mutant Huntingtin to have a clinically significant benefit. Would this be something that you could do in a preventive fashion in individuals prior to the onset of disease to delay the onset of disease? And then there's also the bigger issue of that many of these studies, while they do reduce the mutant Huntingtin, also seem to have some effect on the wild type or normal Huntingtin protein. And we know that the normal Huntingtin protein is a very valuable protein in preventing cell death. And so trying to address all those issues is a challenge. An interesting potential therapeutic approach for neuroprotection in Huntington's disease comes from the Parkinson's disease research community. Recently, Michael Schwarzschild has shown that high levels of uric acid may slow progression of Parkinson's disease. And in fact, this has led to a phase 2 clinical trial funded by the Michael J. Fox Foundation looking at inosine to elevate uric acid levels and hence slow progression of Parkinson's disease. Based on this information, it was thought to look at the CARE-HD study and see if there was any influence of uric acid levels on disease progression in Huntington's disease, and lo and behold, there was. So individuals at the highest levels of uric acid seem to have a slower progression in terms of total functional capacity, as well as a variety of other functional measures. So if this shows to be a fruitful approach in Parkinson's disease, then it might be reasonable to consider similar approaches in Huntington's disease. Obviously, high uric acid levels aren't all good. Complications including gout and kidney stones are a major concern. Still, it's interesting and an area of potential investigation in the future. In conclusion, talking about the future of Huntington's disease research, really we still are at a place where there is a major unmet therapeutic needs in terms of symptomatic treatments for Huntington's disease. While we have one approved therapy for the involuntary movements, chorea in Huntington's, we don't have treatments that actually have been shown to otherwise improve motor function, voluntary motor function in Huntington's disease. Probably even more significant, we haven't identified therapies that might improve the cognitive dysfunction in Huntington's disease. And clearly, cognitive dysfunction is a major cause of functional impairment in this condition and requires a targeted approach to addressing. The identification of neuroprotective treatments, as we mentioned earlier, is the holy grail of Huntington's disease research. Slow disease progression, delay the onset of disease and significant disability, intervening with neuroprotective therapies prior to disease onset, potentially using RNA interference and other techniques to reduce Huntingtin expression or reduce Huntingtin accumulation in the brain. And then the unclear issue of restorative therapies and how do we optimize treatments in this area. What are the role of stem cells in Huntington's disease? Previous studies that we've looked at have really targeted the striatum, but we know that that Huntington's disease is a disease that affects the entire brain, and is that really a feasible approach to treatment? Ultimately, meaningful treatments in Huntington's disease depend on a cooperative community-based public health effort. Huntington's disease is a rare disease. In North America, probably 30,000 individuals are affected with Huntington's disease. Maybe 150,000 individuals are at risk by virtue of having a parent with the disease. Of those, probably 60,000 carry the mutant Huntington gene. In order to identify treatments that slow progression, that delay the onset of illness, we're going to need large numbers of individuals enrolled in clinical trials to do that, and that's going to take a probably worldwide public health effort to do so. Prospective longitudinal follow-up of research participants is going to be critical. In reality, to show that somebody has a delay in onset, you're probably going to need to follow them for 5, 10 years, even longer. While we have some sense when people are expected to develop disease based on their current age and their trinucleotide repeat expansion length, really that is a rough estimate, and there's a wide variability around those estimates. So even if we enroll individuals in a preventive trial looking to delay the onset of illness, we don't have a clear understanding of when they're going to develop disease. So if we're using disease onset as our outcome measure, we're going to need to follow people for long, long periods of time. Probably makes more sense to do this in a staged approach where we look at therapies at their impact on potential markers of pharmacologic activity, such as 8-OHDG. Does it impact the mechanism that we think it's impacting? If it's antioxidant, does it affect 8-OHDG levels? Does it have an impact on other biomarkers, such as imaging biomarkers? And then ultimately, does it result in a slowing of the accumulation of clinical symptoms, the slowing of functional decline? In a preventive trial, does it ultimately result in a delay in the onset of disease? In that same vein, we're going to need an integrated, standardized database of relevant clinical imaging and biological and genetic markers in order to do a staged approach towards identifying meaningful therapies for Huntington's disease. Thank you for your time, and thank you for watching.